Okay. Good evening, Pratham. Good evening, Samish. Okay. Okay. So I'm Rahul Saha, and today we'll be commencing the photosynthesis chapter. Okay. So we had talked about this in a bit earlier. So why is photosynthesis so important, and what is photosynthesis? So the thing is, we have already talked about it. So I'll just reiterate it for you. So photosynthesis is a phenomenon by which the plants are able to use up the light energy. Light energy means the quantum mechanical equation H equal to h nu. Planck's constant is h. You can put, and this energy is used up by the plants. Okay, in the and also carbon dioxide, water is used. I'll tell you how to produce. You can say ATP, NADPH, and in the end, via dark reaction, we are able to produce the starch or sucrose that the equation says glucose. Okay, so the thing is, we had already discussed about the electron transport chain. Good evening, Gayatri. So we had already talked about the electron transport chain. So in this case, what happens is the human beings, okay, the human beings are able to get energy, or you can say produce energy, by a different method. The opposite method is used in plants. Okay, so I'll just do a quick recap for you that why do we eat food is because we want to get electrons from the food. Yes, we want coenzymes, cofactors, vitamins, you need proteins, you need fats, you need lipids and all, nucleic acids, but biochemically we take food in order to get electrons. The electrons will be accepted by the electron acceptor known as NAD plus, which will get converted to NADH and H plus, because it takes two hydrogen ions like this, and it will get convert this into hydride ion, which will have double, and this will be converted to plus double electron. It will become NADH and H plus will be released. Then fad will be converted to flavine adenine dinucleotide to fad H2, and flavine mononucleotide to FMNH2. Right now, after they have accepted, or they have been able to accept nicotinamide adenine dinucleotide hydrogen, it has been produced, then NADH will come through the outer mitochondrial membrane, as the outer mitochondrial membrane is extremely permeable to most of the small molecules and ions. But the story is a little bit different. It's quite, you can say, contradictory in case of the inner mitochondrial membrane. The inner mitochondrial membrane is not even permeable to NADH. Okay, it is heavily or highly selectively permeable. Okay, so NADH, what it does is it can not surpass or cross the inner mitochondrial membrane. It cannot enter the matrix of the mitochondria. Therefore, what it can do is NADH can two NADH molecules can throw one electron each, a couple of electrons, a pair of electrons, and what happens is the standard reduction potential, or the electron gain enthalpy, you can say, of complex 1 will be more than that of NADH, and complex 2 will be more than that of complex 1. Okay, like this complex 4 highest, complex 3, then complex 2, then complex 1. So 1, 2, 3, 4, 5. 5 is F1, F naught, ATPase. So now two electrons, when they come down their concentration gradient, it will lead them to throw 4H plus, okay, out. But again the question arises is the inner mitochondrial membrane is a plasma membrane in the end. It is lipid bilayer. It has got a lipid bilayer, means non-polar. How come or how does H plus is effluxed out? So the answer to that question will come in a little bit, that H plus binds to an intermediate molecule, okay, known as ubiquinone, which is a lipid soluble benzophenone isopenoid ring containing molecule, and it becomes ubiquinol. The double bond O becomes OH, and then H is exported out like that. Okay, I'll show it to you. I'll show the structure to you of ubiquinol also. So it happens like this. Okay, if I show you ubiquinol structure, okay, let's see the structure. Okay, you see, look at this structure. This is ubiquinone. Okay, so once it takes the first electron, it becomes a semiquinone anion or a semiquinone radical. Okay, here it is a semiquinone radical, and then both of this one will become OH. Okay, after accepting one more electron. Okay, then it will become QH2. So this is the completely reduced. You might be knowing that alcohol is the most reduced, whereas carboxylic acid is the most oxidized form. This is how it accepts. You see this double bond O converts, gets converted to OH. This is how it accepts the hydrogen ions, and then it will pump it out and become ubiquinone again when it goes to the intermembrane space or intermembrane side of the inner mitochondrial membrane. It becomes ubiquinol when it is at the matrix side in the inner mitochondrial membrane. That is how H plus is transported out. Okay, then what happens is, here fat H2 will come in. Okay, electrons will come, one pair will throw here 2H plus, then here again 4H plus, here again 2H plus, because 2H plus will become get attached with half O2 and form H2O as the end product of oxidative phosphorylation, because 300 ml of water is also produced at the end of the oxidative phosphorylation as accumulation of all the mitochondria present in all the cells of our body on daily basis. Now after this, you have generated the gradient here. You have been able to generate this gradient. It is known as the electrochemical gradient, okay, which we have talked about a lot of times. Electrochemical gradient. When you have generated this electrochemical gradient, meaning what? H plus has come into the intermembrane space, so positive charges have come there. So already negative deficiency, positive deficiency is there in matrix. So negative charges would prevail, net negativity will come. That is the electrical gradient. And since H plus concentration is directly proportional to the pH value, okay, pH is equal to minus log H plus, thereby more H plus means, suppose the pH becomes here 2, here becomes 5. So now the tendency of physics is to lower the potential energy. It will come down the gradient, return into the matrix through the complex 5. Okay, so H plus will return through the complex 5, and that while returning, potential energy will be released, and that will be harnessed. Okay, it will turn, you can say, 90 degrees each. Okay, binding catalysis, you can say rotational catalysis method, that I'll explain to you later on. And then by harnessing this energy, it's a molecular machine, the F1 F0 ATPase. It will be able to phosphorylate ADP with Pi, form this phosphoanhydride, or you can say covalent bond, and produce ATP. That's how you produce ATP. But the thing to remember or concentrate here is, electron is going down the concentration, down the, you can say, electron affinity gradient. Maximum electron affinity is towards, is by complex 4. And in complex 4 we have oxygen, which has the maximum affinity to accept electron. That is why we need oxygen for breathing out as well. For breathing in and breathing out, you can say. If oxygen could be replaced by any other gas which had more electron gain enthalpy towards electron and would be soluble in the blood and would also bind with hemoglobin with, you can say, appreciable affinity, and would also be present in air so that we don't have to carry that special gas in the cylinder on our back, then we could have replaced oxygen too, right? So the thing is, it goes down the electron affinity gradient. But in case of plants, we are heterotrophic. We are getting food, so we are deriving NADH. Okay, but the plants are not getting food; they are synthesizing food. So in our case, what's happening is NADH, 2 complex 1, 2 complex 2, 2 complex 3, 2 complex 4 means oxygen H2O. You are going down the electron affinity gradient. But in case of plants, how will you get electrons? Okay, the thing is, they are not eating food, they are generating food. So they have water. From water, you have to generate NADH. Okay, so plants generate NADPH, nicotinamide adenine dinucleotide, then phosphate hydrogen. Nicotinamide adenine dinucleotide, phosphate hydrogen, a phosphate is added in the structure. So how are you going to generate NADPH? Because without NADPH, no anabolic products can be made, because anabolic products are reductive in nature and then it can be oxidized. Okay, when some reduction is happening, this can be oxidized, this can be oxidized. Okay, then reduction can happen. Reduction means synthesis. Now, how will you be able to comprehend, or how will the plants be able to go through this reaction? Because they have to go against the concentration gradient. Know it very, very clearly. They have to go against the concentration gradient. So, how will they be able to go against the concentration gradient? Because the tendency to lose electron is more here. So, how will they lose electron in this direction? Because 4 has more electron gain enthalpy than 3. So how will 3 take electron back from 4? That is the greatest question that must be answered. So one way to understand is more why chemical is the, you can say, water is a very, very poor donor of electrons. Its standard reduction potential is only 0.186 or 0.816 volt, you can say. Standard reduction potential of water is 0.816 volt only, okay, compared to minus 320, or you can say minus 0.32 volt for NADH, okay, minus 0.320 volt for NADH that we are talking about, okay. Means that NADH is a good electron donor, a very, very beneficial or a very, very productive or prolific electron donor, whereas water is a very, very poor electron donor, right? So how is it going the other way around? So like this, you need some kind of active transport, because in active transport NAK, ATPase also, you are throwing Na against the concentration gradient, 3Na outside the cell, where outside the cell you have higher concentration of Na plus, and you are taking 2Na plus, you are taking 2K plus in, whereas K plus concentration is much more than its extracellular concentration, so intercellular K plus is much more than the extracellular K plus. So this is your active transport, right? 3Na out, where your Na plus concentration extracellularly is high. Intracellularly is low. Still, you are throwing 3Na out and 2K plus in, where K plus is intracellularly high, extracellularly low compared to its own K plus concentration. So this requires the dissociation of ATP to ADP and PI. This is the dissociation of the gamma phosphate bond to get the energy, and this energy can cause this specific event, flow of ions uphill against the concentration gradient. So here will you dissociate ATP? No. What we do is we go for photophosphorylation in case of plants. In case of animals, it is known as oxidative phosphorylation. In case of plants, it is known as photophosphorylation. Now, photo means the sunlight here is able to provide energy. So you might be asking, we are also standing under the sun. That can we do the same? Can we achieve the same milestone? No, because we do not possess the chloroplast. We do not possess the molecular machine chlorophyll to harness that amount of energy equal to HNO coming directly from the sun. But the plants, they possess that original chloroplast. They have chlorophyll A, chlorophyll B. They have the accessory pigments also, lutein, zeaxanthin, astaxanthin, not astaxanthin, then carotenoids, beta carotenoid, alpha carotenoid, which are the greatest of the machines to harness this energy. I'll explain to you how. And when this energy is harnessed, you have that energy supply, and that extra energy is able to throw this electron or cause the electron traversal from, you can say, higher electron affinity to lower electron affinity, or you can say minus from the plus, you can say positive 0.816 volt to negative 0.32 volt of NADH as standard reduction potential. Okay. Now, this is happening via photons. So, photo, in the end, when the gradient will be established, phosphorylation of ATP will be done. So, photophosphorylation. There, NADH is throwing out or transferring electron to complex 1. NADH is getting oxidized to NAD plus NADH plus H plus. We are becoming NADS, NAD plus. So, that is oxidation. Again, that is forming chemiosmotic gradient or electrochemical gradient, chemiosmotic theory. That is causing, you can say, phosphorylation of ADP, that is oxidative phosphorylation. Here it is photophosphorylation. Okay. Now, how does the plant, or how do the plants cause this specific event, or can cause the perpetuation of, you can say, so many specificities? So the thing is, first of all, let's look at the chloroplast. So we know that the chloroplast of the plants are made up of granule thylakoids, which are disc-like pouches arranged in stacks, and they are connected by stromal thylakoids. Okay, and embedded in the thylakoid membrane. The thylakoid membrane is known as lamellae. Okay, are the photosynthetic pigments and enzyme complexes that carry out the light-dependent reactions and ATP synthesis. Okay, meaning that chloroplast, you can call it a triple membrane, not even double membrane. One membrane and second membrane. Okay. Now these are the granule thylakoid. The membrane are known as lamellae. Here you have a lot of deposition. In the lamellae, you have a lot of deposition of photosynthetic pigments. Okay, like chlorophyll A, chlorophyll B. Then they are connected by so these are granule thylakoids. These are the stromal thylakoids. Okay. Now they are connected by the stromal thylakoids, the stack like granule thylakoids. Okay, and this is the stroma. Okay, stroma, just like the matrix. This is the stroma. Membrane number 1, membrane number 2. You can call this membrane number 3. So chloroplast is technically a triple membrane organelle, not even a double membrane organelle. Okay. Now this is your chloroplast proper. Okay. So that is what in some books it is known as, you can say, stroma lamellae and all. So then chloroplasts are what? They are just one of the several types of plastids which are unique to plants and algae. Now we know that plastids are also self-reproducing organelles. They are bounded by a double membrane, this time a triple membrane, and they contain a very small genome, a circular genome. If you know the endosymbiotic theory, that genome is known as plastome, plastome. Okay, so for neat level, you can write it down. The genome in all the plastids is known as plastome. Now, the plastome can encode some of the proteins produced in the or is present in the plastic, but not all. It is also dependent on the nuclear DNA. Now, amyloplasts are colorless plastids because they lack chlorophyll, okay, and any other chromophores, which are the colored pigments, okay, and they are or they have no internal membranes analogous to the thylakoid membrane of chloroplast, and in plant tissues they are rich in starch, okay. Now, how these plasmids amyloplasts, they pack up the starch granules, okay, so that is there. Now next up is, what we can do is, we can go for the specificities of, you can say, fluorescence and excited state theory, little bit chemistry here. So we know that when a photon is absorbed, okay, just listen to this, an electron in the absorbing molecule, that is the chromophore or the chlorophyll, is lifted to a higher energy level. Now this is like an all or nothing principle, right? Like you must have studied this one, photoelectric effect, where you have this threshold frequency h nu naught, right? Nu naught making threshold energy h nu naught. So E is equal to what? H nu naught plus half mv square. If you have reached h nu naught, then only you can throw the electron from the gaseous atom at the gaseous state from the outermost orbital. Otherwise, you cannot throw. All or null. If you want 1000 hertz, if you have given 999 hertz, still you cannot throw. It will then return to its ground state, right? The electron. So, a molecule that has absorbed a photon here is in an excited state, you can say, and in an excited state is generally unstable. Now an electron lifted to a higher energy orbital usually returns rapidly to its always lower energy orbital, okay. That is, the excited molecule decays to the stable ground state, giving up the absorbed quantum as light or heat, or it may use it to do some kind of chemical work, right? Now, light emission accompanying decay of excited molecules is known as fluorescence. If it comes back to the ground state, from the excited state, now light emission accompanying the decay is called fluorescence. Very important phenomenon, okay. So fluorescence is what? It is always at a longer wavelength or not? It is always at a longer wavelength, that is lower energy than that of the absorbed light, okay. Now an alternative mode of decay central to photosynthesis involves direct transfer of excitation energy from an excited molecule to a neighboring molecule, okay, without fluorescence. Now how is it possible? Getting my point? What I am saying, in fluorescence, in photosynthesis, what happens? Suppose these are the antenna molecules, the sideways accessory chlorophyll molecules. When their electron gets excited, they do not return to the ground state. They'll transfer this energy to the next chlorophyll molecule, exciting the electron. Transfer this energy to the next chlorophyll molecule, exciting the electron, and then it will return to the ground state, but it has transferred the energy. It is not wasting the energy in the form of, you can say, light or heat. How is it possible? Number one, okay. So the thing is, just as the photon in a quantum of light energy Okay, the exciton is a quantum of energy passed from an excited molecule to another molecule in a process called as exciton transfer. So this is happening like an exciton transfer, but how is it happening? Okay, so the thing is, when we talk about chlorophyll, okay, the most important light absorbing pigment in the thylakoid membrane are the chlorophylls. So chlorophylls are what? Green pigments with polycyclic planar structures. Okay, they resemble the protoporphyrin of hemoglobin. Okay, except what? Mg2 plus is at the center, not Fe2 plus. In case of chlorophyll, they do have polycyclic planar structures that have Mg2 plus as the central atom, not Fe2 plus. Very, very important. You can say 1 mark neat question. Even it is asked in home examinations also in school, at school level also. So the thing is, I'll just show you the structure of, you can say, hemoglobin. The protoporphyrin ring of hemoglobin. Okay. So this is the structure of the protoporphyrin ring of hemoglobin. You can see this is the structure. Okay. Of the protoporphyrin ring. If you want to see more, you can look at like this. Okay. Or this one. Okay, this is the structure. Fe2 plus is present at the center. Okay, this one will bind to O2, and each protoporphyrin ring is bound to one of the polypeptide chains because hemoglobin has alpha to beta 2. Each one is attached to, suppose, one protoporphyrin ring to one alpha, another to another alpha, two alphas done. Next one to one beta, another one to one beta. So four protoporphyrin rings attached to individually each polypeptide chain of the hemoglobin. Each protoporphyrin ring, you can see, is able to bind to, it is bound to histidine by this side and four nitrogen atoms, coordination chemistry, and then one oxygen atom. So each hemoglobin molecule can bind to four oxygen molecules, you can say, one oxygen molecule here. So the thing is, this is protoporphyrin ring, and if you see the chlorophyll molecule, chlorophyll structure, so this will look like the same, almost the same. Okay, just look at this one. Structure of chlorophyll. Does it not look like the protoporphyrin? Okay, ring. It is central is Mg2 plus here. Here it is not having any protein alpha beta chains, but the protoporphyrin ring, it is almost the same as that of the, you can say, hemoglobin. And here you can compare chlorophyll A and B. Okay, chlorophyll A and B. This is chlorophyll A, this is chlorophyll B. You can just compare. Okay, chlorophyll A and B's structure. Okay, you can just compare them. This is our chlorophyll A and chlorophyll B. Now, you can say the four inward oriented nitrogen atoms of chlorophyll are coordinated with what? Mg2 plus, and all chlorophylls have a long phytol side chain which are esterified with ester bond to a carboxyl group. Okay, in the fourth ring. Now the heterocyclic 5 ring system that surrounds the Mg2 plus has an extended, also you can say, a polyene structure with alternating single and double bonds that you can see. So chlorophyll or the chloroplasts, they always contain both chlorophyll A and chlorophyll B. But you can say although both are green, right? Their absorption spectra are sufficiently different, means at which wavelength they'll be absorbing maximum light. That is known as the absorption spectra. To check, talk about chloroplasts containing chlorophyll A and chlorophyll B. So their absorption spectra differs. Absorption spectra means the exact wavelength. Okay, 585, 600. What is the wavelength? The exact wavelength at which the maximum absorption of light occurs. Light occurs. That is known as the absorption spectra. So you can think of it as difference in the choice of wavelength. Okay. So in evolution, what has happened is the sunlight has multiple wavelengths. You must have seen the wavelength spectrum. Okay. So visual spectrum also has a range of wavelengths. So evolutionarily, why we have chlorophyll A or why do the plants possess chlorophyll A and B is they complement each other's, you can say, range of light absorption in the visible region. Means suppose chlorophyll A can absorb at 600, it can absorb at 550, like that. So they are complementing each other, okay, in the visible region. Now most plant contains about twice as much chlorophyll A as chlorophyll B. So you can say chlorophyll A 2, chlorophyll B 2 ratio 1, almost, okay, may not be accurately but approximately. Now the pigments in algae, okay, and photosynthetic bacteria also include chlorophylls, but they differ, you can say, only slightly from the plant pigments when structure is, you can say, determined or it is the criterion. Now cyanobacteria and red algae, they employ phycobilins. Okay, cyanobacteria and red algae, chlorophyll-like pigments known as phycobilins. Very, very important. Okay, phycobilins. What kind of phycobilins? Their name is for cyanobacteria. They have phycocyanobacteria, and for red algae they have phycoerythrobilin. Okay, so phycocyanobacteria, they have phycoerythrobilin. Okay, so note down these two pigments. Okay, now phycobilins again are covalently linked to various specific binding proteins. Then what they form is phycobilin plus protein, phycobiliproteins. Now these phycobilins can be attached to various proteins which can give rise to phycobiliproteins. That is there. Now along with the presence of, okay, along with the presence of, you can say, chlorophyll and all, we also have accessory pigments. Okay, accessory pigments, okay, which are of, you can say, equal importance as the chlorophyll. They do not harness the equal to H nu and cause the transfer of electron from H2O to NADS. That is done. That is, I accept. But they are as important, as significant as the chlorophyll itself. Why? So first of all, what are accessory pigments? Okay, the thylakoid membranes, the lamellae, also contain secondary light absorbing pigments known as the accessory pigments. The thylakoid membrane also contain secondary or accessory light absorbing pigments. Okay, now these pigments are referred to as collectively, cumulatively as carotenoids. Okay, you might have heard of beta carotene and all. So they are known as carotenoids. Now carotenoids could be yellow, could be red or purple in color. Yellow, red or purple in color. Okay. So the most important of it is beta carotene. Okay, which is found in carrot also. So the most important is beta carotene. You have alpha carotene also. Beta carotene. Now, which is a red orange pigment. It is again an isopenoid ring, like you can say cholesterol itself, like you can say ubiquinone. Okay, and the yellow carotenoid is the lutein. Yellow one is the lutein. Okay, so this one is red orange. Now, what is their work? Now, the carotenoids absorb light at wavelengths which are not absorbed by the chlorophylls. Okay, so they are also known as supplementary light receptors. They absorb wavelengths which are not absorbed by chlorophylls and are thus called supplementary light receptors. Light receptors. Now, their main function is still to be unveiled. Their main function is they also protect the downstream components or entities from a highly reactive form of oxygen. Okay, like the superoxide oxygen, singlet oxygen that is formed when intense light exceeds the system's capacity to accept electron, that causes photo bleaching. Now I'll tell you what is photo bleaching. Suppose the light is very intense, it is falling on chlorophyll. Chlorophyll is now able to throw out the electron. So if the electrons thrown are too much, okay, too much oxidation happens due to E equal to H nu on chlorophyll, too much oxidation. Too much oxidation is happening. Okay. Now what will happen if too much oxidation happens? What could happen? So the thing is that will lead to more and more electron in the thylakoid. Those electrons can give rise to superoxide radical. That can give rise to oxidation of H2O to produce peroxides. That can give rise to oxidation of OH to make hydroxyl ion. Okay, hydroxyl ion. Or hydroxyl, you can say free radical. It's not an ion, it's a free radical. Hydroxyl free radical. Now, how will this hydroxyl free radical oxygen singlet or superoxide, peroxide cause damage because these are heavily reactive species. They can react with lipids, they can react with DNA, they can react with the plasma membrane of various organelles or the cell itself and cause the cell to undergo necrosis or cell death. So we need to protect them. This is known as oxidative damage. Okay, that is why when you look into the sun or directly onto the sun, your eyes can get photo bleached and you can become blind. And that's why your eyes, the human eyes, also contain two accessory pigments known as lutein, the same lutein which is contained in the plants, and also zeaxanthin is also contained in plants. Okay, so lutein, zeaxanthin, along with beta carotenoid, alpha carotenoid, what they do is they can accept these extra electrons. Okay, they can reduce this to H2O. Okay, catalase enzyme and all superoxide dismutase. Along with that, they can accept these extra electrons from these free radicals. These species are known as free radicals or ROS, reactive oxygen species. You might have heard of a lot of this, that free radical damage, reactive oxygen species. So these are what? These are what? In many advertisements you might have heard, these are nothing but plant-based antioxidants. This is also present in our eyes. They are antioxidants. So they prohibit the oxidation of, you can say, these molecules because they will oxidize and reduce others. So they are antioxidants. So they will do what? They will reduce themselves, not let any other thing to be reduced. So they are the antioxidants and they will protect the plant from the plant cell from death. Okay, so that is why these accessory pigments are so very important because they can quench. They are also known as quenchers, free radical quenchers, oxygen radical quenchers. Okay, so they are also known as quenchers of free radicals or reactive oxygen species. So that is how they function in the plants in the process of photosynthesis. Okay. Now what happens is the chlorophyll, okay, it will funnel the absorbed energy to reaction center by exciton transfer, means transfer of electron from accessory chlorophyll to the main set of chlorophylls in the reaction center known as photochemical reaction center. Okay, so the light absorbing pigments of thylakoid or bacterial membranes are arranged in functional arrays that is given in your book as photosystems. Okay, so the light containing or absorbing pigments, I should be saying, the light absorbing pigments, okay, of thylakoid or bacterial membranes also or bacterial membranes are arranged in functional arrays by, they are referred to as photosystems, called as photosystems. Now, if you talk about a spinac chloroplast, for example, so each photosystem, okay, each photosystem is a congregate. It's a congregation of, you can say, photosynthetic pigments and accessory pigments also. So each photosystem in an spinac chloroplast, okay, in a, you can say, spinac chloroplast Each photosystem consists of 200 chlorophyll and 50 carotenoid molecules. So this is how it is a congregation. 200 chlorophylls and 50 carotenoid molecules. Okay. Now, all the pigment molecules in a photosystem can absorb photons, but again, listen to this thing. Only a few chlorophyll molecules associated with the photochemical reaction center, okay, which is a specialized protein to which the center chlorophylls are attached, are specialized to transduce light into chemical energy. Not all these chlorophyll molecules can convert light energy into chemical energy. Only few chlorophyll molecules as associated with the proteins known as the photochemical reaction center can perform that function of converting your light energy into chemical energy. So I'll write this down. This is a very, very important sentence. You can write it down. All the pigment molecules in a photosystem, all the pigment molecules in a photosystem, okay, can absorb photons or are capable of absorbing photons. Okay, photons is nothing but equal to h nu. Okay, but only a few chlorophyll molecules chlorophyll, I'll just acronym it as CHL, chlorophyll molecules, okay, which are associated with the photochemical reaction center, okay, molecules associated with the photochemical reaction center, okay, are specialized to transduce you can say light energy into chemical energy. Okay, so that is the main sentence that you should be focusing on. The other pigment molecules in that photosystem, they serve as antenna molecules like accessory molecules. Okay, so the other pigment molecules in that photosystem, in that photosystem serve as antenna chlorophylls. Okay, very, very important. Now they absorb light and transmit it rapidly and efficiently to the photochemical reaction center. Okay, now some of them are part of a core complex around the reaction center. Okay, others form the light harvesting complex around the periphery of the core complex. So you can say that chlorophyll and other pigments are always associated with specific binding proteins that I was telling you, photochemical reaction center, which fix the light absorbing molecules like chromophores in relation to each other and to other protein complexes and to the membrane. Okay, so it is like this. I'll just show you the image. It will become very, very clear. Like each monomer of the trimeric light harvesting complex LHC2 contains 7 molecules of chlorophyll A, 5 molecules of chlorophyll B and 2 of lutein. I'll just show you the light harvesting complex. It will become crystal clear. Okay, what do I mean? Just look at this image. I'm just sharing it. Just look at this image. The entire concept will be crystal clear. I think now you will be able to see the image. I think you can see the image, right? Now you can see the image. It's visible for you. Can you see the image properly? So, yeah. So the thing is, in this image, okay, good. So in this image, what you can see is, just concentrate here. These are, okay, the carotenoids and other accessory pigments. Okay, these are the antenna chlorophyll molecules bound to the protein. Okay, and these molecules, they absorb energy and transfer it to the reaction center. You can see this is the photochemical reaction center, which converts the energy of a photon into a separation of charge initiating electron flow. Okay, so these are the antenna chlorophyll molecules. This is the entire photochemical reaction center, you can say. Okay, so this is how that entire thing is, you can say, congregated, and all the antenna chlorophyll molecules will transfer, okay, will transfer the energy which was gained due to the E equal to h nu coming from the sun to the photochemical reaction center, which is again associated with the protein complex. This is the light harvesting complex LHC. Okay, so this is all protein and chlorophyll mixed. Here the specialized proteins, they make this chlorophyll the photochemical reaction center chlorophyll, and only these chlorophylls present in the photochemical reaction center can convert, you can say, the chemical energy, sorry, the light energy into the chemical energy. Okay, ATP and NADP and NADPH later on. Okay, so this is how the reaction proceeds. Okay, so next up, now how does this reaction take place? Okay, number one thing, and why does, okay, the question remains. I'll tell you the question, you can try finding out the answer. Okay, and we'll discuss it in the next class. Okay, so the thing is, just listen to this question. So what I'm asking is, here you have antenna chlorophyll molecule. 3, 4, 1, 2, 3, 4. Here you have the photochemical reaction center. Now what you had here is equal to h nu coming from both the directions. Okay. Now that will cause the electron to reach the excited state, but it's not returning to its ground state and losing energy as heat or light, or causing, you can say, if it lost that energy as light in its decay, it would be fluorescence or heat dissipation. It is not doing so. It is transferring that energy to the next chlorophyll molecule, which is then exciting its electron here. It is then transferring it here, transferring it here. But nowhere are they returning to their ground state and causing fluorescence or heat transfer or heat loss. But when that, you can say, energy comes to this one, this electron in the photochemical reaction center here, now electron will be expelled out. So my question is, why not the electron, after reaching the excited state, they are returning to their ground state? Why don't they return to their ground state. Okay, that is my question. Okay, so that is how the question will the my question is to you, and that is, and the answer of this question I'll be giving it to you in the next class. Okay, so I hope you are able to comprehend the lecture. If you have any doubts, you can contact to me, or you can contact me on my personal number. Okay then.